biologist. Okay. So Nair is a different form of repair. This is named this because it's called nucleotide excision repair. Okay. And when I was learning Nair and Bayer, I kind of hated my life as an undergraduate. And I don't want you guys to hate your life. So I know they sound exactly like the same thing. You're just swapping out nucleotide for base. So I'm going to try to be really clear about what the differences are so you guys sort of have that list in your head. But these are unfortunately named extremely similar processes. They are fairly similar, but they're definitely different mechanisms, okay? So nucleotide excision repair. Again, this can handle many types of mutations. So it can fix.
Is this like prokaryote or
They both have Nair, yeah. But again, it's better understood in prokaryotes. So when I get into the mechanism, that's prokaryotic, yeah. But eukaryotes and prokaryotes have Nair and Bayer. But again, it's better understood in E. coli, yeah. And so many types of mutations can be repaired with nucleotide excision repair, including, again, the photoproducts like thymine dimers, full missing bases, chemically modified bases. This is actually, I would say, in your notes, if you're thinking about what's one of the major repair pathways, the key repair pathways in cells, nucleotide excision repair is one of the most go-to type repair pathways. Okay, it's going to be far overused, even over base excision, mismatch repair, some of these other ones that we're going to get to with all these lovely acronyms that you guys get to memorize. Nucleotide excision is the most common, most used repair pathway. The difference from Bayer that I wanted you guys to have in your notes, because I don't know, for some reason the way in the first class I took on genetics, they were just threw up these examples and I was completely confused about what the difference was. So tell me.
do you mean like chemically bases?
Modified. I mean modified, and I said it in my head and didn't write it down. Yeah. Like the tautomers, whenever we think about the chemical modifications that can happen to the bases that make them an abnormal base. Okay. So this definitely uses different enzymes, which is why it's been named a different pathway and all that from BER. And so these are kind of so I'll give you like two key points in your head. Why do I have to memorize base excision repair and nucleotide excision repair? Nucleotide excision repair uses different enzymes than Bayer, and it tends to remove longer stretches of DNA. So more nucleotides get removed in NER than they do in BER. And you'll see as I write up this mechanism up here, you can definitely tell there's other, we don't use the aglycosylase and all that, but I just wanted you guys, because at least to me the name sounds so familiar, so I wanted you to kind of have these key points. I know key points make us feel safe, like we know what's going on. We've got key points, bullet points. So those are your big differences. Different enzymes, and usually takes out more bases, a larger fragment than base excision does. Okay, so here are some of the proteins that are involved. So this is this is definitely an E. coli. Eukaryotes have a whole different host of enzymes and proteins that they use. Okay. So I'm a super cartoon figure person, visual person, so I'm drawing these as like boxes and squares and rectangles, and I apologize if that's insulting to your intelligence, but this is how I think of things and try to remember it. So they're all named UVR and a letter. So UVRA, UVRB, UVRC, etc. Okay. And the reason that the proteins that are involved in this repair pathway are named this stands for ultraviolet repair. Okay. So when I wrote over here this thymine dimers, this is one of, originally when this mechanism was discovered and characterized, it was discovered repairing photoproduct damage, so UV damage, like thymine dimers. Okay. So they named the proteins after ultraviolet repair, so UVRA, UVRB, there's a UVRC. And the last one that you guys have to know here is UVRD. Okay. And I'm just picking arbitrary symbols and colors because I'm a visual learner. Okay, so this is our list of proteins that are going to be involved in this pathway, okay? And then we can kind of list out what this is going to look like. So if you have your strand of DNA, and if we have something like a UV dimer, so a thymine dimer, we talked about the cyclobutyl rings, they can make the 6,4 dimer product. So whenever you have a thymine and it's bound to another thymine, okay, instead of hydrogen bonding to the A's that are across from it, okay, it makes a big, I'm trying to show this with this terrible drawing, but so it makes a big lump in the DNA. Whenever you take two bases and you hydrogen bond them to their neighbor right next door on the backbone instead of across the way on the other side of the double helix, it bulges the helix out. Okay? It makes a big three-dimensional, what people refer to as a lesion. So when this happens, UVRA, I'm showing it so you can kind of think that it's like one black box, the way I'm showing it here. But this protein operates together with two dimers of this protein. So that it's always found, UVRA is always found in duplicate, okay? And it's going to scan the DNA with the help of UVRB. Okay. So you have two UVRAs and a UVRB. And they recognize those bulky mutations like that. And so this scanning, this is one of those like, okay, we scan the DNA, but when is this actually happening? They tend to scan a lot at transcription. Okay, so they're looking for this stuff to happen at transcription, but they're also just around and floating in the cell. And this is one of those big areas still right now, even as well understood as this is in E. coli, this is a huge area of research in terms of how can you take a few proteins, so there's only, let's see, there's like, what was the figure? I think it was 20 to 50 copies of UVRA and B, at any given time in the cell. And you have a few million bases to scan in an E. coli, and you have 20 to 50 of these proteins floating around. Hopefully those numbers sound terrible to you. You should, I mean, if you think about millions of bases that need to be scanned and checked for damage, that's why this is a huge area of research, because we don't fully understand how this is accurately happens. How can you take 20 to 50 proteins and in any, not taking like weeks and weeks and weeks to do this in a bacteria's lifetime, scan millions of base pairs? And so what they have found by doing this kind of research is there are times when E. coli can ramp up production of these proteins. If it senses that it has a lot of damage or that it's undergoing some kind of UV, for example, exposure, we see upregulation of these genes. All of a sudden, these protein products get produced at high volumes so that we can increase that 20 to 50 copies and really scan the DNA a lot better. Okay. But it's interesting, at least to me, it was always interesting to think about, like, we talk about all these repair pathways, but how is this happening? How is our DNA getting scanned? When is it getting scanned? That kind of stuff. But this heavily happens at transcription in E. coli. Okay. So this is kind of like your first step, UVRA and VR scanning. And what's going to occur is once they recognize that bulky lesion, UVRB will bind. So it's binding directly to the DNA. And this binding of UVRB, so it's in complex with the A's, and when it binds to that lesion site, that binding causes a conformational change such that the UVRAs will fall off, causes them to disassociate. So what happens, so we have A bound. B is bound. So UVRB is hanging out there. The UVR A's have fallen off. And the next step is this binding of B also recruits. So it does two things. It causes UVRA's proteins to fall off, and it recruits in UVRC. So this binding of B to the damage site is going to recruit UVRC. And UVRC is going to sit, the reason I drew it like a long blue hot dog, I guess, is it's going to come in and sit kind of spanning, it pretty much binds to UVRB in the middle of the UVRC protein and spans this area of the DNA quite some distance too. And actually comes up and makes incisions on either side of this damage point. So after UVRC sits, it's going to bind to UVRB, and it makes dual incisions, or dual cuts in the DNA. Okay, so it's going to come up. So it's not a double-stranded break, though. Again, this is only nicking one side of the backbone, not the full double helix. Okay, but it's nicking it on a fairly far distance. This is why that whole, one of the distance differences between Bayer and Nair is that Nair cuts out a larger segment of DNA. Okay. So what you have here, kind of left over, bond there. But you have a segment that includes those thymines that are involved in the dimer here. Okay. You have this little chunk of single-stranded DNA that's been cut off the main helix. So to fully get this apart, I just drew it kind of floating midair there, but we need the help of UVRD. Okay? So after C makes the nick, the cut, D is a helicase. So D acts as a type of helicase, so it's going to chug in here and lift off that single-stranded chunk. So it's going to disassociate, it's going to break those hydrogen bonds that's between the nucleotides. Okay, so essentially UVRD, so it'd be more accurate if you want to like think about this in real time actually happening. Okay, it's chopping off those bonds there. Okay. That are left between that single-stranded chunk. So C's just making a nick, and then there's still hydrogen bond. That ladder kind of structure is still there. So UVRD, the helicase, comes in, opens up the helix, same way a normal helicase works. Okay, only in this case, once it opens it up, this little strand can be totally disassociated. So you have like just a fragment left. Now we're left with a segment where we're missing some bases. But luckily we have a free 3' end and we have a template. So how could we fill this in?
DNA polylipase.
The polymerase, yeah. Even doesn't matter which one at this point, kind of in a sense, right? But it turns out it's DNA pol 1 again. Okay, so DNA polymerase 1 can fill in this section, right? And you'll use a ligase again to connect the last bit. So DNA pol 1 finishes in using that other strand still as a template. You can use ligase at the very end to seal that last, make that last covalent bond. Does that mechanism make sense? Kind of ish. Questions, Sarah?
So the UVRC just makes like little nicks, and then these would actually fix it.
Yeah, UVRC is similar to an endonuclease where it can just make nicks, but it actually has those domains on either end of the protein. So the way it binds and sits to B, it spans it and can cut on at 2 sites versus just 1 site. Yeah. And it's essentially cutting just wherever it happens to fall on the full length of that protein. Yeah. Other questions about nucleotide excision? So there's actually kind of doing a tangent here in our repair pathway explanation. There's a really famous human disease that's associated with mutations in NAIR. So this is called Xeroderma. Xeroderma pigmentosus. So you can see it as XP. This is a human disease. Okay. And it's derma. So derma, what does that make you think of? A disease associated with skin. You guys are awake. You fooled me. Good job. So this is inherited in an autosomal recessive fashion. And so as I just said, it's actually caused by mutations. It's like a backwards thing, because we're talking about repairing mutations, and this is caused by mutations in the NAIR protein. So this is how we explain it for prokaryotes, and this is in eukaryotes, obviously in humans. But the disease itself is when we see mutations in those proteins that are supposed to be repairing mutations, essentially, right? And so typically it's about 8 of the proteins. So 8 or more of the genes that encode proteins used in human nucleotide excision repair. So there's many more. For eukaryotes, there's way more proteins than UBR A, B, C, D. Okay, so aren't you happy I presented prokaryote? So I don't want you guys to have to memorize all the stuff for eukaryotes. It's a similar process though. Okay, so conceptually it's a similar process. This disease, xeroderma, is usually we see it when there's many mutations. I'm not just talking about one of the proteins that's involved in nucleotide excision, it's 8 or more. And so essentially the features of this disease they're a result of a buildup of DNA damage, right? Because if your repair proteins are damaged themselves, if they're mutated themselves, they can't fix DNA damage that's happening, okay? So this is a result of a buildup over time of unrepaired or damaged DNA. And in particular, you guys all picked out that it's derma, so dermal skin stuff. So in particular, this is for UV damage. Because that's one of the major mutations that nucleotide excision repair tends to work on and fix. So these folks have, the patients with XP are extremely sensitive to sunlight. Okay? They're both their skin, their dermal areas, and their eyes themselves. I've had folks, when I've talked about this disease before, say, I'm really sensitive to sunlight. On the level of most of folks who have this disease, it's a really sad statistic. It's like they've had their first skin cancer by the age of 10. Okay, it's extremely sensitive. They almost have to completely cover themselves from the sun, because their cells are unable to fix any of the damage that's being caused by the UV rays. Normally, we can go around and be exposed to sunlight and UV rays, and our cells are able to fix and repair any kind of dimers and things like that. But these folks are heavily prone to skin cancer, as you might imagine, and other problems caused by UV damage to our DNA. So I think it's an interesting disease from the context of what happens when you mutate or damage those entities that are responsible for fixing the damage in the first place, okay? Because then we see a whole buildup of DNA lesions and mutations. I guess before I finish, is there any questions about nucleotide excision? So our next one on our list here is MMR. Okay. What kind of damage do you think mismatch repair fixes? Yay! I asked the easy questions just to see if like sound comes out of your mouth. So it's totally dead for the world. So MMR, mismatches, okay? So anywhere where we see a base paired to an incorrect base. In particular, these are mutations that polymerase should have fixed. Okay, so this is like the cleanup after polymerase screwed up. Okay, so this is going to fix those mutations, which are mismatches. So polymerase should have found during what? What step or type of action? Yeah. So polymerase has a directional proofreading, right? 3' to 5', it can proofread. So during its 3' to 5' proofreading step, it's supposed to fix and find those mismatches. But there are a certain percentage of times that it does not. So then mismatch repair proteins have to come in and repair those kind of errors. So again, this is another sort of form of redundancy, right, by the cell. The cell has a 3' to 5' proofreading by polymerase. If that fail-safe doesn't work, then mismatch comes in and tries to fix it. So it has to sort of, I'm breaking this one down by the steps that have to occur for this to happen, because the second step introduces a whole new sort of concept to what's happening to DNA in prokaryotes. Okay, so we'll get to that. So step one, you have to recognize this system, this mismatch repair mechanism, has to recognize a mismatched base. And step two, it has to actually, so first it has to recognize that there is a mismatch. Then it has to determine which of those bases is the correct original base, right? Because we've talked about that before when we were talking about types of mutations. One of the issues is which one's correct. Okay? If you have a T paired to a G, you as a repair protein have to know, is the G the one that's supposed to be there, or is the T the one that's supposed to be there? So it has to determine which is the original or correct base. Or another way, I guess I could say it backwards here. So which base is the incorrect one? And this is the point, you know, like a little star on your nose. That's the point we'll come back to. How it's able to do that is inherent to this mismatch repair system, but it's also inherent to certain mechanisms in bacteria. Okay? And eukaryotes work a little differently, but we're focused, again, this is work done in E. coli to help explain. So then three, of course, we just take out the incorrect base. And then fix it, so fill in with correct bases. So for this number 2 point, so how does it know which is the correct base? So it turns out that mismatch repair, again, is we're kind of thinking about polymerase's job and how he failed and sucks at life, and so now we have to have mismatch repair come in and do his job for him. So polymerase is acting at replication. So this is during S phase of the cell cycle, right? So these kinds of errors, these are replication errors, right? they're gonna produce a mismatch on which strand? The new or the template?
The new.
Yeah. Exactly. So the newly synthesized strand is the one that's going to have a problem on it. So as we're undergoing replication, when we see a problem, incorrect base stuck in, this repair system has to essentially, here, this determining which base is the incorrect one. Really what we're saying is this repair mechanism, these proteins have to distinguish between template, original strand, and newly synthesized strand somehow. There has to be a marker on it that shows this one's template, this one's newly synthesized. And so what bacterium do is they methylate their DNA. So this is the reason I didn't really introduce methylation and bacterium and prokaryotes before. We just talked about it in terms of imprinting, epigenetics, silencing, and eukaryotes. Okay? And I didn't want you guys to get confused thinking methylation always means silencing, because in bacteria it doesn't. Okay? But bacterium, they methylate their DNA. So this is a normal process for them. It doesn't silence the gene, and they methylate certain segments. So they're looking for adenines. They're specifically looking for adenines, and they're looking for adenines only in the following sequence. So anywhere along the bacterial chromosomal DNA that the sequence is GATC, they stick a methyl group on the A. So if you took out normal bacterium DNA chromosomes, everywhere there's a GATC, there's a methyl group hanging on that A. That's a normal state for bacterium. It doesn't mean that that area is silent. So that's very different from eukaryotic methylation. But what happens so in relation to what we're thinking about for mismatch repair, there's a lag time right after replication where we have a newly synthesized strand that has not been methylated yet. Okay, because you have to have an enzyme that comes in and adds methyl groups if that's its job. So right after replication, the DNA is considered, it's called hemimethylated. So instead of both strands, because GATC is a palindrome, right? So both sides, both strands, the A is going to have a methyl group on it. But directly after replication, only the old strand has a methylation. Because there's a lag time, there's a delay. Specifically before the adenine methylase gets there. So this is how this mismatch repair can use the methylation patterns to distinguish between old and new synthesized DNA. So it can tell the difference by simply looking for these little methyl groups hanging off the adenines, whether this is a new strand or an old strand. But it has to use, you guys can kind of see, it has to use that window of time, right? Because instantly if adenine methylase comes in, starts adding methyl groups, then the mismatch repair system's kind of screwed in terms of telling which is different. So I want you to kind of just make a star in your note. That's where we're going to come back to on Wednesday. We'll stop right here for tonight, and we'll come back and finish describing what mismatch repair is doing, using this way to distinguish between old and new strands. And I don't, so one question before you guys all start shuffling around.
Yeah, just, could you repeat one more time? You said this is how mid-strand repair recognizes what?
Between, template original strand DNA and newly synthesized DNA.
Okay, then we'll go on to.
Yeah.
what's after that.
Yeah. Yeah.
In the random reset replication error for one million synthesized DNA, that's before it gets repaired, right?
Right. That's talking about how these errors are generated, yeah. Okay, so I don't mind if you guys shuffle and